Okay, welcome everyone to the Linacre seminars, which are virtual at the moment, and we're just going to be waiting a few minutes for other people to join, so bear with us for a moment. I'm Samantha Vanderslot, I'm a JRF at Linacre, and I've been running these seminars with Gail Preston.
Hello everybody.
Okay. And for those of you who have joined, you'll note that you can't speak, but you can enter into the meeting chat if you have questions along the way. And then there'll be about 10 minutes or so after the presentations for those questions to be answered and discussed. So please do enter into the chat as we go along, and we'll also prompt you to enter questions after the talk.
I don't know, should we start
Yeah, let's start. A few more people have joined. So first up, we've got Duncan Murdock, and he's a research fellow actually at the Oxford University Museum of Natural History. He's going to be going first. He's also a JRF at Linacre, and his work has been on the fossil record and how to use that to understand early evolution. So he's going to be talking a bit more about well, the Cambrian explosion and the first vertebrate skeletons. So on to you, Duncan.
Right, thanks very much, Samantha. Can everybody hear me? Yep, I'm going to assume I've got nods. Great. Yeah, so thanks for that introduction. So I'm Duncan. So I wanted to start off by with some thanks. So there's a whole host of people that have contributed to my research over the last 3 years. So I've been in Oxford at the museum for 3.5 years, and coming up to the end of my JRF in Linacre. So I also wanted to say thanks to everyone at Linacre College for making me feel so welcome and for helping in all the ways that they have. Because I'll be in September, I'll be moving to a new role, still in the museum in Oxford, but as a collections manager rather than in a research focused role. So I'll try and talk for the next 50-20 minutes or so about the research that I've been carrying out here in Oxford. So first of all, there's I've got two different hats to wear, virtual hats. First of all, I am an evolutionary biologist. I'm interested in the way that different animals are related to each other in deep time. And so you'll see in this talk a series of different trees. So these are different ways of representing those relationships. Here's a you are here arrow. In this case, it's we're being represented by Charles Darwin. So these lines just connect animals that share a common ancestor, more closely related to them than any other creature in the tree. The other thing that I am is a geologist. So I always like to think of things in terms of their time. So we have a representation here of geological time, and the period that I'm going to focus on is around this red line that you can see here, the major boundary between the so-called Ediacaran or Precambrian and the Cambrian, where we see typical kinds of fossils today. So first of all, this diagram is a little bit wrong because the scale on it is a little bit skewed. So this is the same diagram rescaled to show you all of Earth history on the same scale. So for the vast majority of time, there was very few recognizable organisms that have reached the fossil record, nothing complex like an animal. And then we reach this red line and everything happens over the subsequent 500 million years or so. So it's this event in Earth history that I want to talk about. It's often known, as the Cambrian explosion. So if we zoom in on that red line, which is around 541 million years ago, these are the two periods of time that we need to become familiar with. The name the Ediacaran and the Cambrian, named after the Ediacaran is named after the Ediacara Hills in South Australia, where rocks of that age outcrop, and the Cambrian is named after the Cambria, which means Wales, because in parts of North Wales and parts of Southwest Wales we get Cambrian rocks exposed. So here's a reconstruction of what we think this ocean was like in the Ediacaran, in this example from a site of exceptional preservation in Newfoundland in Canada. So there are creatures that may or may not be animals. The debate is still raging on that one, but they are weird and wonderful. They don't look very much like the creatures that we see today. If we fast forward just a few tens of millions of years into the Cambrian, so here is a reconstruction from another site of exceptional preservation, this time in Yunnan province in China. We see again lots of creatures that are unfamiliar, but basically they are very similar to the kinds of animals that we see today. They have heads and eyes, they have legs, they're eating each other, they're living in the kinds of ways that animals in the oceans live today. So we have this very extraordinary change that takes place in the in this time period, where we get the evolution of a whole host of different creatures. Now, if we break this down a little and have a look at a time in between these two events, so this is right at the end of the Ediacaran, around 543 million years ago, we still have fossil evidence for these strange creatures, the remnants of the Ediacaran, but we start to see more recognizable Cambrian-style animals. So they're here on this image. And the crucial thing is that these have hard skeletons. They're using minerals to build skeletons, and it totally changed the game. And I want to tell you a little bit more about how and why that took place. So first of all, why are skeletons important? So animals today use skeletons a variety of different ways. When you think of a skeleton, you probably initially think of something like this, but the diversity of skeletons are much greater than those of vertebrates. So here is just a handful of different animal skeletons. Some of them are on the outside of the body, some of them are on the inside, and they're made from a variety of different materials: calcium phosphate, silica, like this sponge here, essentially made of glass, and calcium phosphate, like our teeth and bones, and like this, the tooth here of a shark. And skeletons are extremely widespread. So this is an evolutionary tree. It's essentially the same as the one I showed you at the start with Darwin, so we are here in this case. And I've added on where we see skeletons or biomineralization. That's animals using minerals in their bodies, and it's all the way across the animal tree of life. And animals use skeletons for a whole host of different things. So corals, like the ones over here, actually change their environments. They build whole ecosystems out of their skeletons. Some animals, like this bivalve, the geoduck, they build a shell in which to live. And many animals use their skeletons to capture prey or to avoid predation, like these two animals here. So what exactly happened, and when did it happen? What took place during this the so-called Cambrian explosion? So here is the red line that you saw in some of the earlier slides, just oriented in the other direction. And we've got deeper in time over here. We see these branching points represent when the major different groups of animals split off from each other. And these took place These green bars represent the range of confidence we have on these dates, and they all happened deeper in time, in the Cryogenian or the Ediacaran. Then we cross over the Cambrian boundary, and very shortly afterwards we get fossils of almost every kind of animal that we see today. So this is really what the Cambrian explosion is about. It's this significant increase in the number and type of different animal fossils that we see. So this is just the same diagram really, but expanded a little to include a greater variety of different kinds of creatures. And there's a chart superimposed on this tree. In blue are the so-called animal phyla. So these are the major different kinds of things like arthropods and vertebrates, segmented worms. And then within those are the classes. So that's things like mammals, fish, spiders, etc. And again, we see this huge and rapid increase in the numbers of different kinds of animals we see during this time interval. So zooming in again on this very important period of time to try and see what actually took place. So down here we've got these weird soft-bodied Ediacaran animals, and then as we cross into the boundary we get this rapid increase in diversity of different kinds of animals. So to give you some examples, here are these strange soft-bodied Ediacaran creatures. All you need to know about these for the purposes of my talk is that they don't have any hard skeletons. And then this is an example of one of the first animals with a skeleton. It's a thing called Narmacalaphus. We don't really know what it is, but in this cross section through this strange rattle-like animal, this here in bright white are original minerals that the animal built its wall of its shell from. And here is another example of one of the first biomineralizers. Here is a creature called Cloudina. It was probably something like a coral today. They lived in these little stacked cones of mineral. And these early animals started to build the very first examples of reefs. And then as we get further into the Cambrian, we get the so-called small shelly fossils and this huge explosion in diversity of different kinds of organisms. So all of these are from the very early Cambrian, a whole host of different animal groups, and they come from all across the animal tree of life. So we have vertebrates that I'll going to talk about for the remainder of the talk, but echinoderms, the relatives of starfish and sea urchins. We have cnidarians, that's jellyfish and corals, and we have various different kinds of worms, little mollusk shells like this beautiful example here that looks just like a tiny little snail. Brachiopods, which today are very rare, but any of you who are fossil collectors will have come across a brachiopods, I'm sure, and arthropods, that's spiders, insects, crabs and their relatives. And the complexity of the environments that these animals lived in was able to increase due to the building of skeletons. So here we have some early sponges and corals actually building the environment in which they lived. And ultimately we get this kind of diversity of ecosystem with animals doing a whole host of different kinds of things, including the very first vertebrates. So these little fish-like things swimming into the corner of the image are some of the very first vertebrates that we see in the fossil record. So why did the skeleton evolve when it did? Well, it isn't really a single cause. It's really a Goldilocks effect of a whole host of different things coming together. And this very busy diagram is just to get across there's a whole range of different factors. But for the second half of my talk, I'm just going to focus on two things. So in the middle here, we have some animals begin to mineralize their skeletons. And here we have genetic complexity of animals increasing. And we have this feedback between how complex animals are in the way that they code for building their bodies and the origin of more and more complex skeletons. So to take a case study, I'm going to look at the group to which we belong. So when you think of vertebrates, you probably think of mammals or even fish, like this amazing skeleton of a of a tuna that we have in the museum. But the vertebrates that I'm going to talk about are much more primitive and simple than that. So I'll introduce them to you in a moment. But first of all, some genetics. So there's this piece of work from a few years ago now. The details you don't need to worry about, but what I want to point out is that they examined the genes that are responsible for building skeletons in sea urchins that are one of our nearest invertebrate relatives, and those invertebrates, and they found although they don't share a skeleton from a common ancestor, they do share genes for building those skeletons. They called this a common bimerization toolkit. So this is thought to have given rise in vertebrates to different kinds of more and more complex skeletal tissues in more and more complex vertebrates. So to try and understand this a little bit more, we need to look what's going on right at the base of the vertebrate tree. So this little creature here is called Amphioxus. It's in the group of called cephalochordates. They're one of the very closest groups of invertebrates to us. And the thing I want to draw your attention to is here labeled this is collagen. So this is a large biological molecule that's used as the precursor for skeletons, like a scaffold. So this is direct evidence from modern animals that there was the scaffold for an organic skeleton and the molecules that you need to build skeletons in deep ancestors. So we can have a look at the fossil record of the earliest vertebrates. So this is a tree of some of the very earliest vertebrates and the range of different bony tissues that they possessed. And there is a whole host of different things going on. So we have this stepwise accumulation of more and more complex tissues leading to the group that all of modern vertebrates derive from. But this is only really looking at one example. So I wanted to find a sort of independent test of do we see the same pattern in different groups of vertebrates. So for that, I turn to my pet group of fossils. So this is a pinhead with 4 different fossils mounted on the top. These are all from a group of creatures called conodonts. So they were small eel-like fish that had these mineralized teeth-like elements in their mouths and that they used to catch and capture their prey. So this is where they sit in the vertebrate tree of life. So we are down here. We're just a derived version of a lobe-finned fish. So we fall into this group here. And these are all of the rest of the vertebrates that most people recognize. And then over here we have the jawless vertebrates, the hagfish and the lampreys, and sitting in between them are these conodonts. So they arise sometime in the late Cambrian and they went extinct at the end of the Triassic. And so we know what we know about conodonts because of a few exceptional finds of their soft tissue. So here is muscles of a conodont preserved with its teeth-like elements that form these arrays of sets of teeth that ultimately you can reconstruct into these apparatuses that they use to capture their prey. But the very earliest conodonts, which are the ones that I'm interested in, they had much more simple teeth-like elements. They were essentially just little cones. So here is 100 microns for scale, so that's 100 thousandths of a millimeter. So they're very, very small fossils, and they're just these little, very simple cones. And they come in 2 broad different types, and we can differentiate them based on how that they grew. So we have the so-called paraconodonts that have a single tissue represented here in blue, and the euconodonts that have 2 tissues in their in these little teeth. They have this capping crown layer. And we know from the fossil record that these are totally independent from the rest of vertebrate skeletons. So we can use them as a test to see, do we see the same kind of patterns in conodonts as we see in other vertebrates. So here we have the growth of a paraconodont compared to a euconodont. This is just to show that hopefully you can see if this in this video. There we go. That euconodonts are just paraconodonts that you've added an extra bit of tissue onto. Otherwise, they're exactly the same. I don't think this is going to play, so I'll move on. So the paraconodonts form this group of creatures down at the base of the conodont tree. So I wanted to examine what they look like on the inside. And just like in the rest of vertebrates, we see this progressive acquisition of more and more complex tissues. So we start off with very, very simple forms with just little cones that just have this cone in cone structure. And then we get more complex tissues and ultimately the development of this crown tissue at the top, which is very similar to, for example, enamel that we see in teeth. So going back to this diagram to finish, so we see in vertebrates that their soft-bodied ancestors had the capacity to build skeletons that they inherited from their last common ancestor. So they had an organic scaffold onto which they could build their skeletons, and then as their complexity of the genes associated with building those skeletons was developed in the early part of the Cambrian, their skeletons became more and more complex. And this had huge knock-on effects for the diversity of animal ecosystems. So, and vertebrates are just one example. So I could have bored you all evening by going through all of the different animal groups. So I've found exactly the same pattern in mollusks, so things like snails and squid, and the same patterns we see in corals and jellyfish. We see the same pattern in sponges that have skeletons, where we have their ancestors are entirely soft-bodied, but with an organic skeleton scaffold, and then they build more and more complex genes and more complex skeletons as a result. So I'll leave you with a quick advert. So as part of my role in the museum as a research fellow, I was also able to contribute to an exhibition. Unfortunately, for obvious reasons, you can't go and see this exhibition for real, although it is still there in the museum, and hopefully we will open up again before it closes. But we have all of the content of this exhibition is on our website. So the address here, oum.ox.ac.uk/first animal. So as well as having all the exhibition content, we also have a range of talks that are taking place. There's one tomorrow by my colleague, Dr. Imran Rahman. So there's details of all of those on our website as well that you can find out more about this topic if you're interested. And I'll leave it there. Thanks very much for your attention.
Thank you very much, Duncan. Some fantastic images there. I mean, I think it really brought it to life. How did you collect, such a nice range of pictures, and are they from the university collection? Are they from the museum? Yeah, so a lot of these are specimens that we have in our collections, or specimens that I've brought with me, and most of the way that I image these tiny fossils, so they're all less than a millimeter in size, so they're very difficult to see what's inside. So I use a technique called synchrotron tomography. So we take these to an X-ray source. In the one I have used in the past is in Switzerland. Expose these fossils to X-rays, and you get a 3D rendering of the entire internal anatomy of these little fossils. So that's what allows you to reconstruct what's happening at a very, very fine scale inside. So it's really fun work to do, but it's quite painstaking with these working with these very small creatures. Yeah, very good. So we're gonna get some Q&As now. If anyone wants to ask a question, you can type it into the Q&A box on the side. So does anyone have anything they want to ask? Also, from Gail, do you have any questions also?
Yeah.
At the moment we don't have any.
With your, I mean, obviously it's always very speculative, but with the very tiny conodonts with their very tiny teeth, what do you think their very tiny teeth were for? What was the purpose of their teeth? Yeah, that's a really great question, and
yeah, so that's one of the other sort of research streams that I've involved in, is trying to understand the function of these things. So we've done virtual experiments where you can stress these little elements by applying a force at the tip and seeing how they respond. And we found that some of the different shapes are much better at resisting bending in one direction than the other. So exactly the same as a blade. So these were almost certainly used as cutting tools. So lots of them have, although they're cones, they're flattened in one direction with a sharp edge on one side. So they were probably used to actually capture and process prey items. So all of this is taking place on a very small scale, but there are lots of marine predators today that are of a similar size and scale that prey on zooplankton and other small planktonic organisms. So we think that's probably what conodonts were doing. But they are a hugely diverse group of fossils. So there are thousands of different species that we see with a huge range of different morphologies. So their diets were probably as diverse as fish are today. So they probably did a whole host of different things. But yeah, we think the very earliest ones are almost certainly predators living near the sea floor and preying on microscopic older organisms.
At the moment we don't. Oh, okay. We've got a question. Were their teeth innervated?
Yes. So the two different tissues. The outer one is hypermineralized, so it's almost entirely solid mineral with no soft tissues in it at all. But there is evidence that the other tissue in the base was connected to soft tissues. Now, what they were, whether they were blood cells or whether they were nerves, as you've asked, is up for grabs. One of the problems that we don't really understand about conodonts is that vertebrate teeth grow by emerging from the soft tissues, and they're still connected at the base, and you don't get more growth on the cutting surface, on the outer surface. But in conodonts, we have direct evidence that they were broken and then repaired. So the only way to do that would be to have soft tissue on the outside of these elements. So the prevailing idea is that they were able to be retracted back into the soft tissues, and then more mineral laid over the top. So they must have had a connection. So there must have been some sort of nervous connection to make that kind of decision, as it were, to repair broken elements. But very little is known about them, because although we have thousands and thousands, or probably millions of conodont fossils, there are only 12 in the world that contain any evidence of the soft tissues at all. And they're only from 3 different species. So we know very little about their soft anatomy. Yeah. Thanks.
So if there aren't any other questions, Gail, should we move to the next?
Yeah, thank you very much.
Center?
Yeah, thanks everyone.
Thank you, Duncan. Okay, so we're gonna put on Nikita's presentation.
Hi, can everyone hear and see?
Okay, just bear with us a minute.
Sure.
That should come up now. Okay. Looks like your presentation's up now. Great.
Okay.
So, just very quickly. So Nikita, she's well, like me, a EPA cephalosporin junior research fellow at Linacre, and she's got an interest in diabetes and complications with diabetes, and she's in the Department of Physiology, Anatomy and Genetics. And she's going to be talking about maternal diabetes causing birth defects. So on to you, Nikita.
Thank you, and thank you for inviting me. So, as already said, I'm going to discuss my research and how maternal diabetes causes birth defects. So first, let me talk about diabetes. So diabetes mellitus, you've probably heard of it, type 1, type 2 diabetes. So very briefly, type 1 diabetes is typically diagnosed in early adolescence and is an autoimmune disease and is characterized by immune cell mediated destruction of your insulin producing beta cells. So these are your pancreatic beta cells that produce insulin, and as a result, you get a loss of insulin secretion, and thus these patients need lifelong insulin therapy. Then there's type 2 diabetes, which is typically diagnosed much later in life, around your 40s, although much earlier nowadays, and in particular those with a high BMI greater than 30. And this is characterized by spontaneous beta cell death and reduction in insulin secretion and a reduction in insulin sensitivity. And I won't go into the details, but a very short summary is that obesity and physical inactivity in an individual increases your insulin resistance, increases the stress on your pancreas, reduces your beta cell function, and therefore you get a decline in insulin secretion. And these patients will typically have an oral anti-hyperglycemic, so that's a pill that reduces your hyperglycemia, your elevated blood sugar level, but eventually many of these patients don't recover well or don't fare well and will require insulin therapy as well. Now, there are more than 420 million individuals around the world suffering from diabetes. And whilst the disease itself is debilitating, what actually affects quality of life more are the complications associated with diabetes. And you may be very familiar with them, but to name a few, you have an increased risk of cardiovascular disease, blindness, kidney disease, and that's just a few of them. But what is really overlooked is being pregnant during diabetes and the consequences of that. So what do I mean by maternal diabetes? Well, either I mean that the mother is diabetic at the time of conception. And in the UK in 2015, 6% of pregnancies were to diabetic women. Or she develops diabetes during pregnancy, and this is known as gestational diabetes, and affects between 3 to 10% of pregnancies according to which country you're in. Now, why is this important? Well, diabetic women have an increased likelihood of miscarriage by up to 25%. And clinical guidelines advise diabetic women against getting pregnant, but don't really explain why, which is, again, staggering. Now, 1 in 3 women with diabetes are of reproductive age, so this is problematic for their outcome. And around 60% of live births had some form of hyperglycemia involved. And 1 in 7 births are affected by gestational diabetes. So I know there's a lot of stats to throw at you, but it'll become clear why this is so important. So gestational diabetes itself has no single diagnostic criteria, but is generally accepted that it's a carbohydrate intolerance of varying degrees with first onset or recognition during pregnancy, and will typically disappear or treat itself once you've had the baby. Now, there are lots of risk factors, but I'm not going to go into all of them. But what's quite frustrating is that 40% of cases have an unknown origin, and it's usually diagnosed in the 2nd and 3rd trimester of pregnancy. Now, I know this is a very over-the-top schematic, but the things that I want you to take away from this are that in gestational diabetes, there are pre-pregnancy risk factors such as pre-existing obesity, or the mother has a hyper-inflammatory status. She might have had another health issue prior to that that's caused this status. And as a result, or she might have a predisposition to it. As a result of a decline in insulin production, you have increased glucose production, decreased glucose uptake, leaving the mother to be hyperglycemic. And the short-term consequences for the mother, she might have pregnancy complications, which are devastating. Long-term consequences, she may develop diabetes later in life. But what I'm most interested in are the consequences for the fetus beyond the fetus developing diabetes as an adult later in life. And what I'm most interested in is that infants of diabetic mothers have an increased likelihood of developing a birth defect by up to 5-fold. And these defects include impaired growth. So actually infants of diabetic mothers are much larger than infants from healthy pregnancies. They have neural tube defects such as spina bifida, where there's non-closure of the vertebrae and the spinal cord pouches out, and anencephaly, where parts of the skull and brain are not formed, so this would not be compatible with life. Orofacial defects such as cleft lip and cleft palate, and congenital heart defects. Now, organogenesis, so that is the formation of the embryonic fetal organs, is complete in about week 8 in the human pregnancy. Now, why is this important? Gestational diabetes is diagnosed at 23 weeks of pregnancy. So by the time a mother might find out that she has gestational diabetes, the fetal organs are already formed and perhaps the damage caused is irreversible. Furthermore, 40% of pregnancies are unintended, and this includes the countries in the West, which means that even if you are a mother with pre-existing diabetes and you have relatively well control of your blood sugar, you may not know to have extra tight control of your blood sugar until well into your second trimester. And then again, it might be too late. So this is where I come in. So I have developed a novel mouse model of gestational diabetes using the beta-V59M mouse. I won't go in too much into the details, but using this genetic manipulation, we can control the timing and severity of diabetes in mice. And we activate our transgene using tamoxifen. Once we inject tamoxifen, we switch off insulin secretion. And the beauty of our model, relative to other mouse models, because many other people have worked on this, is that our mouse model requires no insulin supplementation. Our mice have no weight loss. Our mice show no loss of libido, no unrelated sickness. And additionally, this mouse model displays hyperglycemia only. It doesn't display the other complications that come with diabetes, such as protein dysfunction or lipid dysfunction, which means we can really say it is hyperglycemia that causes XYZ, unlike other models. So the aim of my project is to induce hyperglycemia in these mice, to collect their embryos and examine their heart and other organ morphology, under the hypothesis that pre-gestational diabetes or gestational diabetes causes complications in the fetus as a result of hyperglycemia alone. So using this unique model that I said, we'll switch off insulin secretion. 3 weeks later, we'll mate them with a healthy male, and we wait 3 weeks so that they have quite an acute level of diabetes. We then measure the mother's blood glucose, and then I categorize them according to their blood sugar level. And a subset of the severely diabetic mothers will get glibenclamide. So what I also haven't mentioned is that our model is exquisite in the sense that we can also switch insulin secretion off and on when we like. By giving glibenclamide, we can switch insulin secretion back on. No other model can do this. They can merely insulin supplement. We will then collect embryos and placenta at 15.5 days post-conception, or embryonic day 15.5. This is around the same time organogenesis is complete in the human, so it's around 8 weeks of the human pregnancy. We then fix these embryos and we image them using micromagnetic resonance imaging. So the first thing I was able to do with my model, unlike others, is that according to how much tamoxifen I deliver, how much I can control how much of the transgene I activate, and therefore how diabetic each mother is. So as you can see here, we have very disparate levels of diabetes. Now you might think, well, people aren't categorized like that, but actually I think this more accurately represents the human population, because you do get a vast range of people with different severities of diabetes, and by binning them like this, we may be able to tease out more information. With other models of diabetes, you just give your drug and then you see what happens. The first phenotype I observed with my model is that with increasing severity of maternal diabetes was the increased likelihood that the mother wasn't pregnant upon dissection. As you can see here, in the severely diabetic mice, 22 of them were not pregnant, which is staggering. Now, this could relate back to the statistic I gave you earlier that, you know, up to 25% of diabetic pregnancies end in miscarriage. And to that, litter viability was also severely reduced. So again, with increasing severity of diabetes, we had an increased number of dying or dead embryos. Again, this could link to the high termination rate, spontaneous termination rate. So to summarize, with increased diabetes severity, we have increased maternal complications. What about the embryo? Well, what's interesting is that when we measured embryonic blood glucose, they followed a similar trend to their mothers. So moderate embryos came from moderate pregnancies, highly diabetic embryos came from highly diabetic pregnancies, and so forth. They seem to mirror their maternal blood glucose. Now, you might think, well, that's pretty obvious, but that hasn't been looked into thus far. No one has measured embryonic blood glucose in the mouse just yet. And what reflects that is that you have an increased number of abnormal embryos with increased severity of maternal diabetes. And of those that are abnormal, they have a higher blood glucose than those that are normal, i.e. those that have a macroscopic defect versus those that have no defect. So it seems here that embryonic defect might be linked with embryonic glucose as opposed to anything else. So with increasing severity of maternal diabetes, we have increased embryonic blood glucose levels and therefore increased embryonic abnormality. Now, we've used micro-MRI to observe what microscopic defects are caused by diabetes. So here we have a healthy embryo at E15.5 and a healthy sagittal MRI. Typically, we can see delayed growth and improper brain development, as you can see here. Brain is not properly developed, the embryo is much smaller. The digits are also much smaller. We have severe subcutaneous edema, you can see right here and here in the MRI. We have lymphatic cysts as well here. You can see there's no cyst here, there is one here. We also have perturbed eye development. So in this embryo, we have microphthalmia, so there is a tiny eye there, and it's not been able to come out there. And we also can get anophthalmia, so no eye at all. And we get exencephaly. So this is the precursor to anencephaly. So the brain grows out of the head, but eventually dies out. You can see here. Actually, also, you might be able to see there is a small eye developing right there. And you see congenital heart defects. So here we have hematoxin and eosin image of a ventricular septal defect, and we see it here in the MRI. Now, we're most interested in these congenital heart defects, particularly structural heart defects. So these are formed in utero. And the most common ones are, as I mentioned, a ventricular septal defect, so where you have a hole in the heart in the ventricular septum, so the wall between your two ventricles. So here you get mixing of oxygenated and deoxygenated blood, and patients that suffer from these may require surgery and therapy, and if without it, they might be okay, but they might be very easily fatigued and have other health consequences. And another common one are atrial septal defects, so a hole in the wall between your two atria, the chambers above the ventricles. Now this is slightly different. Again, you'll see similar issues, but you'll have more blood being delivered to your lungs, and these patients typically can suffer from heart failure and cardiomyopathy and other severe defects. And these are just a small tight subsection of the heart defects you see. We see a huge range. Now, I wanted to see if I could recapitulate the defects seen clinically, because I'm starting a brand new model. And I could see a membranous VSD. You'll have to take my word for it for some of these, and an atrial septal defect. But I also have to confirm these defects with imaging. So I collect the hearts out, I section them, and then I stain them using hematoxin and eosin. And here we have a control right here, nice healthy control. We have a membranous ventricular septal defect, a muscular ventricular septal defects. The difference between these two is that the hole is at the top of the septum, and this one, the hole is in the middle of the septum. And here we have a very thin wall, myocardial wall, and we have an atrioventricular septal defect. So all of these defects we can recapitulate. So this was quite encouraging. We also see a concept of different ones. So as I mentioned, we have a thinning myocardial wall here. So the heart wall is much thinner compared to controls, you can see here. The area of the ventricles are much smaller. So here we have a 3D render of the heart from MRI. And the right and left ventricles are both smaller in the diabetic heart compared to controls. The yellow here is a ventricular septal defect that I've highlighted. And what's also very interesting is that when we were to stain the coronary vasculature using PCAM, you can see in the control you have a very nice vascular network, but in the diabetics you have these endothelial blisters or blood islands. So in concert, these defects actually are quite problematic. They indicate that not only is the heart not developing well and not forming properly, but its vasculature that supplies it is also not forming properly. And again, which this makes it incompatible with life and possibly may contribute to the staggering spontaneous termination rate. And just to show it graphically, with increased severity of diabetes, we have increased abnormal hearts. So you are seeing a trend here. Now, as I mentioned earlier, my model is completely reversible. So we want to see, could we reverse these defects? So I got when in a subset of severely diabetic or control pregnant mice, I gave them glibenclamide and I embedded it in the scruff of their necks. We waited 2 days, I measured their blood glucose, and then at E15.5, as with the others, I collected the embryos and I scanned them. Now, developing this treatment paradigm was complicated because as hypoglycemia is problematic for the pregnancy, so is hypoglycemia, so is reduced blood glucose. So we didn't want to overdo it. So I gave a very low dosage of glibenclamide in the mother, 0.25 mg per pellet. And as you can see here, we weren't able to reverse anything. This embryo still looks very poorly. Its organs are growing outside of its body. It has exencephaly, amongst other things. So we increased the dose to 25 mg per pellet. Now it seems here the embryo looks much more normal, but it has very poor vasculature and it has no eye. It has anophthalmia. So again, we thought, okay, we're in the right direction. Perhaps we are treating the mothers too late. Treating the mothers at 7.5 days post-conception, that's about halfway into organogenesis. Let's dial it back a bit. So we treated at E3.5, so that's 3.5 days post-conception. That equates to 2 weeks into the human pregnancy. Now, as you can see here, it looks completely normal. Now, whilst that's a success on our part, that means that for very diabetic pregnancies, 2 weeks is the maximum time limit to treat. Now, many women don't even know they're pregnant at 2 weeks. So this is something that we're trying to work on. So obviously we can prevent some of these defects from happening, but what about the blood glucose? So maternal blood glucose was reversed completely, as you can see here, and embryonic blood glucose was also reversed and also brought down. However, there were 4 anomalies here that I've highlighted, but this is very encouraging that, you know, we treat the mums respond rapidly, and the embryos also respond. And perhaps that's why they're not seeing defects. And then when we look at the percentage embryos, we see here that the number of embryos that were reversed, only 4 were abnormal and only 4 were non-surviving. And of those, that were abnormal, they had a higher blood glucose. So again, it seems that embryonic abnormality and blood glucose is linked. And with the other comparisons, when we look at pregnancy comparison, we had a greater number of pregnancies, successful pregnancies, and a greater number of surviving embryos. So all in all, it seems that treating the mums 3 days into pregnancy was able to rescue most of the phenotypes, but not completely. We're still getting abnormal embryos. We're still getting not successful pregnancies. So we still have a lot more work to do here. Heart defects were mostly reversed. Again, we still saw 3 embryos with defects, but they weren't major heart defects. As you can see here, this heart has no defect, but the ventricles are still a little small. And we also briefly looked at the placentas. So placental health, as you may be aware, has substantial influence on embryonic development. This crude schematic that I've drawn will explain it a little bit. But all in all, the placenta is the site of materno-fetal nutrient transfer. And so if it's compromised, of course, the embryo will be compromised. So the diabetic placenta improperly stores sugars. That's shown here, where you see lots of dark purple. And also it's much smaller. It has a compromised maternal and fetal vasculature, which may also contribute to some of the defects we see. When we treat the placentas, they almost look completely normal. So again, it seems like there is a lot to be teased out with treating the mums very early. So to summarize, elevated blood glucose in the mother also induces elevated blood glucose in embryos. High maternal blood glucose halfway into pregnancy is indicative of birth defects in the embryo or even no pregnancy at all. And it may seem obvious, but tight glycemic control in the periconceptual period by diabetic mothers may reduce the prevalence of birth defects in their offspring and also increase their chance of conception. I showed in my slides, but I didn't discuss it, that glibenclamide, so treated control animals, they show no difference in their MRI or their blood glucose compared to those that are treated and compared to controls. So it could be potential therapy for gestational diabetes and their infants. So the next steps of my project involve delineating the molecular mechanisms behind defect formation. This is no mean feat. It's taken me 2.5 years to build this model so far. It'll probably take me another 2 years to tease out why this is happening. And I also want to reverse these pregnancies at different time points, because I want to determine the optimum therapeutic window in terms of public health. I think giving a 2-week deadline is too short. I want to see how far I can push it. But bearing in mind, I've been doing this in the most severely diabetic pregnancies, which we wouldn't really see clinically. I'm going to look at the more clinically relevant types of diabetes and see what their upper time limit is, so that I can again provide perhaps more efficacious public health guidelines. So with that, I'd like to thank my funders, my PIs, and everyone who helped with this project, and yourselves for listening. Thank you.
Great, thank you very much, Nikita. So it looks like we have a question. Why does increased fetal blood sugar concentration cause developmental defects? Is it down to the change in osmotic pressure, causing strange fluid cell flows, for example? I'll just pick that up.
So that's a very good question. The simple answer is, at the moment, we don't know. It could be we haven't really looked at the osmotic relationship. We're mostly interested in the developmental dynamics, looking at the developmental pathways that are affected by this. For example, the defects that we typically see are heart and neural tube defects. So we think that neural crest cells are perturbed, but it could be that the elevated fetal blood sugar, at that point embryonic or blastocyst, well, they don't have blood, but their sort of sugar concentration is so high that it could be causing strange fluid cell dynamics. It's good food for thought.
So I actually had a question. I wondered, this sounds like very, very new research. Are there any other groups doing similar things? And you said about also your next project, you'd hopefully want to look at the upper time limit. Like, how would you go about testing that?
So there are lots of groups doing this. Well, I say lots, there are many, but they don't have access to the model that I have. Most groups tend to use this model of diabetes called streptozotocin, where you inject this drug into the mouse directly, and it essentially obliterates the pancreas. Now, it's not to be too damning of it, but it's a quick and dirty way of doing it. I've used this model before in my PhD. It's great. It's cheap, it's effective. However, this drug itself can cross the placenta and it can cause defects irrespective of the effect it has on blood sugar levels. And so a lot of the work that's been put out there, I'm not saying isn't valid, and I'm not saying it isn't accurate, but as I said earlier, these mice from the STZ model, the strepsitosin model, their mice typically have a loss of libido. They have other uncharacterized sickness. They lose weight very rapidly. They need insulin supplementation to survive. Otherwise, they just kind of keel over and die. So all these other health problems must add as a confounding factor in your analysis. I can't see how it wouldn't. And so I'm not saying I built a perfect model, but mine doesn't really have all these issues. And so that's why my stuff is very new and it's ruffling a few feathers in the community.
That's good.
Oh, and your second question, how do I aim to find the last time point in other pregnancies? Well, as I mentioned earlier, I can create very specific types of diabetes according to how much of my drug I deliver, how much of the transgene I activate. And I can pretty much, prior to even injecting, I can tell exactly what kind of blood glucose range I'm going to get. And so, like I said, I've only been working in the severely diabetic mice. I'm going to work in the high and the moderately diabetic mice and see if I can, because even those animals get embryos with defects, even those embryos have heart defects, which is quite worrying. That this would indicate that a woman with good control of her diabetes during pregnancy could still have an infant with a heart defect, which is obviously very shocking. So I want to look at these more clinically relevant levels, reverse those at different time points in pregnancy. So I'll have a cohort of embryos reversed at say E7.5, E8, E9, and see how that translates to the human pregnancy, because I think 2 weeks is ridiculous.
Thank you very much. Are there other questions? Yeah, I hope you get to carry on that research. It sounds like it's only just started.
Yes, me too.
Okay. Oh, we've got Gail. Come on the screen.
It looks like all the questions we have now.
Okay, so I think we're going to wrap up there. Thank you very much to our presenters. I think you had lots of really great imagery and was something that worked quite well over Teams. So thank you for trying this out with us, and also for everyone who attended. We can't see or hear you, but I've seen that you're there. So thank you all for watching. And we're going to have, well, more presentations in 2 weeks' time, kind of the same format. And we're going to be advertising that soon as well. So watch this space.
Yeah, thank you all for joining us. And thank you very much to our speakers.
Thank you.
Yeah, thanks a lot.
Okay, great.
Okay. And we just had 2 people saying thank you. Oh, and do you plan on exploring the genetics? This is to Nikita, so I want a final question got in.
Yes, I do. So I've actually just sent off some data for RNA-seq to look at the transcriptomic changes in these embryos. I've microdissected out the very early heart and the very early brain, and hopefully I'll get some interesting data back. Well, I've got the data, it's with our bioinformatician, and hopefully something interesting will come out of it. But yes, that's what we're looking at.
Brilliant. Well, I think we're closed there. 2 last questions got in, but yeah, people saying thank you, and yeah, thanks again from us.
Thank you.
Thanks a lot, then.
Bye.
Great.